your PowerPoints. Now, my PowerPoints for the first chapter it kind of matched. For the second chapter and onward, I have my own photos because I was looking at the photos that the book gives. They seem more complicated. Okay? Material is the same, but you will notice that the photos don't correlate. Stuff is the same. And you need to be aware of, if I present you a cell this way, and if I in lecture I present this way, in test I present you a different way, in real life you present a different way, you have to be able to use that knowledge. You cannot be so square. Does that make sense? Okay? Stuff is the same, just presented in a different way. So, if you have issues with me not actually using the textbook photos, I'm sorry, it just makes it more complicated to me. I found it easier once to understand. You always have your book to reference back. Does that make sense? This is why make sure you have my PowerPoint, not Dr. Yusuf, because listen up. Thank you. Because Dr. Yusuf's PowerPoints are exactly what the book gave. Okay? They're basically what the instructor's manuals are. That's what they are. My PowerPoints are custom made. I basically read the chapters, I filtered out the stuff, I made key points, stuff like that. This is why I use my PowerPoints, it's easier. I don't want to be up here and looking at the whole thing. Okay, what am I supposed to talk about this slide? In my stuff, I know what I want to say in there. Does that make sense? Okay. Now, one thing, I don't know why for section 1B, yesterday night I had saved the critical thinking, I had uploaded all the PowerPoints. For some reason, eCollege did not save it properly. I just found that out from a student. So 1A, everything worked perfectly. 1B did it, nothing saved, so I have to redo stuff. Today when I get home, I will work on it, okay? So just hang in there. Okay, can you please turn on the recording? Oh, you did, okay. Okay, so now we're going to talk about basically the cell. What are the components of a cell? This corresponds to chapter 2. Okay, so let's talk about cell doctrine. Basically, cell doctrine is that all things are composed of cell and cell components. Okay, there's trillions of cells in our body. Now, single cell is the smallest unit and exhibits all the characteristics of life. Another rule is that all cells come from pre-existing cells. Okay? Every single cell comes from pre-existing cells. And now there are two different main categories of cells. Okay? You have prokaryotic. Okay, prokaryotic and eukaryotic. Prokaryotic and eukaryotic cells. Prokaryotic cells are basically bacteria. Okay? Does that make sense? We don't touch about that. That you have microbiology, where you study microorganisms. But there are two different classes of cells. Prokaryotic cells and then eukaryotic cells. We are eukaryotic cells. Okay? And we're going to just focus on eukaryotic cells. Now, cytology, as I said, was study of tissue. Okay? Cytology is a study of tissues. Common techniques used: light microscope, transmission electron microscope, or scanning electron microscope. Okay, there are three different microscopes that you can use: light, transmission, electron. Light microscope, magnification is up to 100 times, sometimes 200 max, and basically you see the whole structure. Okay, you're trying to find the structures. Then you have transmission, it magnifies more than light microscope. Then you have scanning. Now this shows three-dimensional images. This is basically a structure called cilia. As you can see, it actually gives you appearance where it's 3D. Where light microscopy and transmission microscopy, they do not give you a 3D appearance, only scanning does. Now, these are different cells in our body, and as you can see, this slide is just to show you the comparison of the size. Okay? This is an egg, a female egg. This is a sperm. You can see the size difference. This is a neuron, which is a brain cell. You could see this is a bone cell. What is the size difference? Okay, and structurally they look different. They look different because they have different function. Their structure allows them to perform a specific function. If your heart is abnormally shaped, it's going to cause problems. If your cells if they don't look the way they're supposed to, it's going to cause problems. Okay? Now, make sure you guys understand this chapter. If I have to tell you what are the chapters you got to focus on compared to the 4 chapters we're covering today, 2 and 3. 2 and 3. This one and the next one. After that is number 4 and then number 1. Okay, number 1 is least important. That's why I went very fast. This one and the next chapter are the most important. I might not have time to cover the 4th chapter, but those handouts have everything written. Just memorize it. And if I don't have time to lecture something, I will try to make those questions simpler compared to the point where I did. Does that make sense? Okay, now, when you look at a eukaryotic cell, basically, inside the cell, it's 2 main compartments. You have a cytoplasm, and then you have a plasma membrane. Okay? Plasma membrane is basically the outer covering. Example, if this is a cell, okay? The walls would be the plasma membrane. Everything inside would be the cytoplasm. Now, the cytoplasm has cytosol and it has organelles. Cytosol is fluid, where organelles are little structures. Each of them have a different function. Does that make sense? So you would say that this classroom is a cell. The walls become the plasma membrane. Each of you are organelles, and the desk, everything else is a cytosol. Okay? You might have, do clinical thinking 1. You might have the lab reports too. Your function might be to summarize chapter 10. Does that make sense? Every single organelle has a different function, and combined, they make the cell survive, and in turn, the organism survive, or the tissue survive. Make sense? Now we have non-membranous organelles, and we have membrane organelles. Membrane organelles, they have a membrane covering it. Where non-one, they don't have a membrane. Okay? Now, this is a classification. We're going to talk about each organelle more in detail. Do you guys understand the concept right now? Yes. Have you guys heard of organelles before? Yes? So it will go much quicker. Now this is a eukaryotic cell showing you the organelles. Okay? And we're going to talk about each one in detail. I just love this one. Now let's talk about the plasma membrane. It encloses the cell. So if a plasma membrane encloses the cell, what do you think the function of plasma membrane is? Protection. Good. Protection. Okay. It's semi-permeable. What does it mean that the plasma membrane is semi-permeable? It's specific to what it allows in and out. Okay. Selective in what it allows in and out. Okay. Function of the cell membrane is protection. Because it doesn't let the good stuff to escape that easily. It doesn't let the bad stuff to come in easily. Okay. Composition. What is composition of a plasma membrane? It's said to be a phospholipid bilayer. Phospholipid bilayer, meaning there are 2 layers of phospholipid. So one molecule of phospholipid looks like this. Okay, that's one molecule of phospholipid. You have a head and you have a tail. Okay, so one layer is like this. Okay, and then you have the second layer. You see, it's a phospholipid bilayer. First layer, second layer. make sense? I see some confused faces. Yes.
I guess the drawing, I'm a little confused. Are they together?
Yes.
Or,
So if this is one phospholipid, this is second set, they're attached.
okay.
Okay. Does that make sense? So right here, you can see the two layers coming apart. This is one layer, and then here is a second layer, right? It's like a zipper, right? You know the two zipper has two layers, and they're closed up. That's how it is. Make sense? Now, phospholipid is a lipid. This is why it's called, because we have different types of lipids. It's a phospholipid. Phospholipid bilayer. So two layers of phospholipids bound together. Okay? Now, between the phospholipids, you see other structures right here. Those are cholesterol molecules. So embedded within the phospholipid bilayer, you have cholesterol molecules. The function of the cholesterol molecule is to give it some stability. Okay? Phospholipid is a lipid. It's just basically very loose. So cholesterol is there to give it some sort of rigidity, to sustain its shape. Okay? To give it stability. Does that make sense? So that is the function of cholesterol in there. So phospholipid bilayer with cholesterol embedded inside it. Okay? Another substance that you see these molecules right here. This right here, this. These are membrane proteins or integral proteins. Okay? Look at the room right here. Look at the walls around you. Besides the walls, what else do you see? Doors. What's in the back? Windows, right? So think of that solid wall as a phospholipid bilayer, and think of the doors and the windows as integral proteins. What is the function of those windows and doors? To let stuff in and out. That is the function of these integral proteins, okay? These integral proteins will allow substance to come in and substance to go out. This is how it's selective. Make sense? Okay? Does the door randomly open? No. It's open when it's time. You guys were here at 7:55. I still wasn't here. Were you allowed to come in? No. You guys came in when I was allowed to come in, or when I came, right? Are you freely able to go? Yeah, you can. But if you leave like an hour later, are you going to get participation points? No. Same thing. So the integral proteins are there to allow substance to go in and out. Again, in physiology, you learn more in detail. Right now, this is enough. So plasma membrane is this phospholipid bilayer with cholesterol and integral proteins embedded within it. Okay? Any questions? Now, if I use images in my test, the images will come from my lecture notes. Okay? I don't try to trick you. The only way I try to trick you is give you clinical case scenarios. That's the only place where I become tricky. Okay? But I will hopefully we will figure out a way to, make your brains learn how to decode clinical scenarios. Okay? Now, there are three transport systems which are used by the body. The first one is, well, not first one, but one of them is passive transport. Passive transport. Second is active transport. Okay. Now, in passive transport, what is the difference? Passive transport, no energy required. Okay? No energy is required in passive transport because solute is moving from high concentration to low concentration. Make sense? So passive transport is no energy required because solute is moving from high concentration to low concentration. Now we have two different types of passive transport. We have diffusion osmosis. We have two different types of passive transport, osmosis and diffusion. Okay? Passive transport. No energy is required because solid is going from higher solid concentration into an area of lower solid concentration. Okay? It's either leaving the cell or entering the cell. But wherever it enters, wherever it enters, the solid concentration is low. Make sense? Okay. Oh, the types were supposed to be osmosis and diffusion. Sorry, I wrote it at the wrong place. Now, active transport. Energy required because solute is moving from low concentration to high. Okay? Now, so active and passive transport are complete opposite. Okay? Passive, no energy required. Active, energy required. Why is passive no energy? Because solid is going from high to low. Where in active, it's going from low to high. Make sense? You have two types. I'm going to erase this. Now, the reason my handwriting becomes a little sloppy is because every time I lift, if it's the same word, it's not aligned. As you can pinpoint exactly where you're going to write, mine doesn't do that. So sometimes if it's the same word, I try not to lift my finger or my hand. Does that make sense? And that's why, because then I will write, the next letter will either come on top of it or too far. Only when it's actually, that's why you'll see it's sometimes trippy. Okay? And this is why I tend to repeat myself. Now, and plus, I've been said I have a doctor's handwriting. So, okay, two types. Primary active transport and secondary active transport. Okay? So active transport, two types, primary and secondary. Now let me ask, are there any students in here who went to a community college before? You guys did. Okay, so you might understand this scenario more. What happens if you were the first registration block to sign? It's your registration date, class is empty, what did you do? You just logged on and registered. You were in the class, right? Now what if this is the last class you required to graduate, to get your associates, but you are the last registration date, or you are the last block. Now it's your turn, all of the classes are full, but you need that class to graduate. Basically, what do you do? Okay, but you are on the waiting list, but what do you do besides that?
Try to add the class.
How do you try to add the class?
Stand in class and hope someone doesn't show up.
So, you basically come to class when you're not supposed to, right? Students hate coming to class. Now you're coming in when you're not in a class. Don't you email the teacher, I really need this class. If I don't graduate, I'm going to be postponed 1 year, and etc. Okay? Aren't you investing energy? A person who was the first registration, they didn't do anything. They went on a computer, signed in, they were in the class. They basically enjoyed the rest of the summer off. Where if you're on a waiting list, you're checking every day. Okay, did somebody drop? Did somebody drop? Why can't somebody drop? I need this class, right? You're actively investing energy. So think of it where if the classroom is full, okay, there's a lot of students in here, not that many out there, so you're trying to go from an area of low concentration to an area of high concentration. That's active, and this is why you're investing energy. When in a passive, this classroom is empty, you just come in. You don't put any energy. Does that make sense? Okay, now the third type is endocytosis, or exocytosis. Endocytosis or exocytosis. it's basically a process where bunch of material is being transported at the same time via forming vesicles. Think of vesicles as little packages, parcels. Okay? So in this one, okay, in endocytosis, substance is coming into cell. Exocytosis, substance is leaving the cell. And both processes utilizes vesicles, which are like little packages. So right here, look at the screen. Okay. You have this vesicle. Vesicle is basically as a package. You have a box, and inside the box you have a content that you want to ship, right? Same thing. These are the contents. This is basically the box. So it's called a vesicle. Vesicle migrates to the plasma membrane. Okay? A plasma membrane, regardless if it's of a nucleus, regardless if it's a vesicle, it's always phospholipid bilayer. So the vesicle comes and it attaches to the plasma membrane. And once it attaches to the plasma membrane, it becomes part of the plasma membrane, and the substance is released outside. Okay? Does that make sense? So is that exocytosis or endocytosis? Exocytosis. Now, in endocytosis, anything that needs to come inside the cell, it will try to aggregate, okay, a clump up around the plasma membrane. So when it starts clumping up, the plasma membrane starts to invaginate. As you can see, it starts to invaginate, and it invaginates so much, then basically you pinch off. And it becomes a vesicle now, which is inside the cell. Make sense? Okay, now cytoskeleton. Think of cytoskeleton as poles to a tent. Have you ever gone camping? And if you want to set up the tent, what do you do first? Don't you put those poles and then you lay the fabric over, right? What was the purpose of this poles? Stretch.
Hold it down.
To basically hold the fabric up, right? Right? Stretching, but structure, right? So cytoskeleton is the poles to a tent. Its function is to give structural support, give a structure to the cell. Structural support. Okay? It's a poles to a tent. Now, there are three different components of the cytoskeleton. You have microtubules. You have microtubules, microfilaments, and intermediate filaments. So right here you could see microfilaments and intermediate filaments. And right here you could see the microtubule. They are attached to one another and as well attached to the plasma membrane, therefore keeping the plasma membrane up or shape. Does that make sense? Okay. So cytoskeleton, poles to a tent, structural support, three components. And microtubules, microfilaments, and intermediate filaments, all of them are proteins. Now you have, do you have cilia flagella next? Or are they hidden? Okay. Now, cilia flagella and the microvilli. Cilia are these structures right here. They're little hair-like structures that are found on the surface of a cell. Okay? Am I missing any slides? No, that was only the, okay. Cilia is on the surface of the cell, and it's like little hairs. Think of it, it acts like a broom. What is the function of a broom? Sweep stuff along. Okay, so cilia sweep stuff along. What does it sweep? Maybe mucus? Okay? sweep stuff along. Location. So its function is to sweep stuff along. Example is mucus. So if the function of it is to sweep mucus, where do you think you find cilia? What system? Respiratory. Don't you cough up some mucus, especially when you're sick? It's because the cilia in your respiratory tract tend to sweep it up and you cough it out. Where else do you think you would find cilia, which has nothing to do with mucus? Where